infrastructure. I'm Lloyd Sumner, and I'll be your host. Behind the scenes are Dan Bearden and Sudanika Ray, and our discussion leaders for today are Drs. Oliver Fiehn and Pieter Dorrestein. Fiehn, excuse me, Oliver. After so many years, I should have it down correctly. So, with that, again, my apologies, Oliver. This is our third in a series of presentations, and again, today we're going to talk about developing and enabling metabolomics databases and computational infrastructure, and we will continue with this series approximately once a month. So just a few comments on logistics. Please sign in using your full name. That's so we can know who's online, and then during the question answer periods that we can call on you by name. If you have technical questions, please use the chat window, and that's down here, and one of us behind the scenes will try to answer your technical questions, i.e. how do I log in, or if you're having problems with audio or visual, we'll try to help you there. In the attendees window, you have an area to enter questions. So if you'll enter your scientific questions here, we will address them at the end of the presentation. It's also helpful if you will raise your hand in that same window. So the idea is that we will have first an oral presentation by Oliver and Pieter, and then we'll go into the a poll time and then do some discussions. So again, our goal is to do a presentation to stimulate thought. Then we want to open this up for public feedback. Historically, we plan for about an hour, but historically we've gone longer than that. So at about 1.5 hours, if we go that long, I'll try to bring things to a close. But our goals today are to really talk about these challenges and vet them and use your feedback to help us prioritize these challenges and related to database development and computational infrastructure. And through this, we hope to build a consensus of what these challenges are, and then provide a network for interactions among our attendees to start building solutions for those. And so again, our discussion leaders today are Professor Oliver Fiehn, who is the director of the West Coast Metabolomics Center. He's housed there at the UC Davis Genome Center, and he will be joined by Dr. Pieter Dorrestein, who's also from California at the University of California, San Diego. He is the director of the Collaborative Mass Spectrometry Innovation Center, and also the co-director of the Institute for Metabolomics Medicine. And so with that, gentlemen, I'll hand it over to you. Pieter, I will make you the presenter so that we can see your slides, and ask you and Oliver to go ahead and take off once you see your slides. Okay.
So you should see my slides at this point? Very good. Okay.
Dan, do you see them? Because I see them on a different screen, just to make sure before we move forward.
Oops.
I see your slides, but
Slide is up.
So they should be okay now.
Okay.
First of all, thank you for having me part of this webinar series, and also thank you for anybody who is listening. I think it's really important in metabolomics to address the need for databases and computational infrastructure. And really, if you think about the goal of these databases and computational infrastructure is really converting the M over Z signals that we get to usable knowledge, and there's a lot of steps in between that actually are pretty challenging, and we're going to highlight what sort of the current status is and where we're going to go. One of the things that metabolomics really needs to overcome is this great metabolomics count anomaly, and that is for every molecule that we can say something intelligent about in terms of structure component, there's typically 50 to 100,000 features or molecules, or however you want to quantify that. So usually about 50 to 100,000 unknown features that can be detected. And so I think using databases and new computational approaches will improve this number significantly. And so, Oliver, go from there.
So the idea in the past is that many people have published papers about databases. They have published papers about how to go from MZ values to usable knowledge or use literature information. But usually these databases and repositories have been isolated. And if you could, somebody click. Okay, thank you. So the idea is we can only conquer the metabolome together. There is not a single repository that will do all of it. And only if we're able to share our data in an open source and open access way, then we can together do this. Now, the important point is that I don't think a single repository like the globe in the middle will be needed, but and I don't think that all repositories have to be, linked to everything else. There can be nodes in between where one repository is copied by the other, and then somebody else links in somewhere else with other types of information. But eventually, we can only do it together if we are able to share also the codes and share this information, maybe through GUIs or other types of information. Okay. So the idea is in the past that people have focused on processing data from raw files to some sort of MZ tables. And these were different types of algorithms and tools, and this has been pretty well established. But now that we have these processed data tables and we have lots of mass spectra, we really need databases to convert this mass spectra to identified or annotated metabolites using libraries and other tools, and that is a huge bottleneck still today. And once we have solved that, then of course we can go forward from identified tables or formats towards information about pathways or background information, diseases, whatever our biological questions really are. The point in the middle is really missing. Next slide.
So I think if you look at some of the purpose for some of these repositories, so if you want to go store libraries and do knowledge mining, there's two components to it. Of course, you need to have the database so you can begin to infer and think about sharing this kind of knowledge, but ultimately you need to build the computational infrastructure. And really, you can separate those out in many ways. So you can use databases to capture and share metabolomics knowledge, to share the data, to make chemical knowledge accessible, to associate metadata with the chemical knowledge. This is repository, and then you need to have the computational infrastructure that allow you to reveal biology, and really also you can build infrastructure that allow you to determine the quality of knowledge, and we need to be able to accommodate novice users of the knowledge, as a clinician wanting to treat patients, to really expert in analysis. So if we explore the type of databases that are available, there's really two types, and those are databases that store reference spectra. So these are spectra of known molecules that later on you can match to, or there are metabolomic databases where you can deposit your entire data set, and of course funding bodies are beginning to mandate this, but there's very few of those places where you can really deposit large-scale data, and I might have missed some of these. But in terms of reference spectra, there's very few of these, or about half of them that I am really aware of, and there are some others as well that are not listed here. There's about half of them that actually make the MSMS libraries accessible to the rest of the community. Others allow you to search their data, but not download the reference spectra, meaning that you can people who want to do computational method development for new algorithms that use known data as references cannot use those particular infrastructures. One thing that is actually really surprising is we've been talking for years that we need to make large metabolomics data publicly accessible. If I look at the dominant repositories for metabolomics, these are Metabolites and Metabolomics Workbench, in total we have less than 350 studies that are in there. More recently, we have just submitted a paper on global naturopathic social molecular networking where we have 230 studies, but that means less than 560 studies are accessible in the public domain. so one of the grand challenges I see is that we need to increase our reference libraries, because right now, if you look, there's maybe 300,000 reference spectra available, and these are only actual experimental data that represent roughly 20,000 to 30,000 molecules, individual molecules. And yet, if you go to PubChem or ChemSpider, there's about 77 to 75 million entries, and so we're not even close to covering all chemical space that has been reported in libraries. And so I think we will be able to overcome that. One example of this is of a repository where you can upload spectra is Mass Bank North America, and Oliver can talk more about this.
So the Mass Bank of North America is following this idea by starting to incorporating publicly available mass spectra into one repository. So starting with that idea that Mass Bank had originally originated in Japan, but then contributing also looking at other places, almost like a crawler, like a web crawler. Whenever something pops up that is available, we would be able to put it into that repository with the idea that there's lots of metadata so that people can search, they can tag queries, they can then have downloads based on their individual instrument or on their, just like for accurate mass, they may want to ignore nominal mass data, or they say, I want all data. So far, we have 33 contributors, and the idea is that anyone could also upload. We have over 200,000 mass spectra that are not unique, and we have now made a unique hashtag for spectra that we can say we have about 100,000 unique mass spectra available for about 70,000 unique compounds. And what we did is we not only have now experimental MSMS spectra or electron ionization mass spectra, but also we allow and tag virtual mass spectra, that is predicted mass spectra. I'll talk more about that later.
So one of the things that is coming very clear is that there's a variety of different repositories for MSMS libraries, and here I'm just highlighting GMPS, NIST, and Mass Bank. One of the key things that I'm starting to see a trend of is if you look through those different libraries, is that they really cover different ground. So here's an example of the total density of MS, the parent mass at least, of the MSMS that were deposited in these three different libraries. And you can see that some libraries are slightly better in higher M/Z than the other. And you can build sort of quantitative information on that. And what you will really explore is that these libraries cover different chemical space. And so and that means the way Oliver described earlier with all these different databases that exist, they need to be cross-linked and cross-communicate much the way Mona is doing at the moment, because that's how we are going to capture the maximum amount of chemical knowledge that exists in these kind of databases.
So you can also think about the other way. We have lots of different instruments. And the bottleneck is compound identification. And for the compound identification, you can say, well, first of all, I would like to match my MSMS spectrum to spectra of known compounds. That's, of course, something we all want to do. But as Pieter Dorrestein will tell you, is also you could also match your MSMS spectrum to networks of other spectra, even if these have not assigned molecular structures at this point, but at least it tells you what kind of molecular types these are, and you would have clusters of these types, and that can be helpful already if there's even one or two compounds in a cluster. You get an idea of what kind of types of molecules you look at. So what Mass Bank of North America currently contains, next slide is, next, yeah, okay. So, go back. Go back. Thank you. There was this kind of pop-up. So right now we have used the data from HMDB. There are some spectra in HMDB. There are spectra from GNPS, that is Pieter Dorrestein's database, where there are assigned structures to it. Then, of course, we have used all the mass bank spectra, and we have also used all the GCMS spectra that we have accumulated over time. These are all experimental spectra, usually from authentic compounds, but then we also included now spectra from virtual libraries like the Lipid Blast library that itself has over 200,000 compounds. Spectra. Next slide.
So just one more comment. I think this is really critical as we move forward in the metabolomics community that we develop infrastructure that can indeed grab the different knowledge out of the different databases. And I think that's really one of the fundamental key differences that's beginning to evolve now in the metabolomics community, that the sharing is happening and that these links are being made. But of course, one of the key features is how we process data.
So as I said before, raw data processing is on the way, and we have done this before. Very importantly, of course, people have to understand that if they publish data, they should also publish data about the study design and how they process the data. And that field is, not very well developed. I mean, there are ways to do it, good laboratory practice, LIMS systems, and of course using quality controls and randomizations. But very often these things are not very well captured in current public studies. Pieter mentioned there are 500 studies public, and you won't find always all that type of information. Then, of course, you go from raw data and you do some sort of peak finding, deconvolution, either in data-dependent or independent data acquisitions in MSMS. And there are different software versions, MZMine2, XCMS, and we published this year MS-Dial to help with that as well, including the mass spectral deconvolution. So these problems have been fairly well covered, but then, of course, you need the compound identification and the annotation, including the quantification, and that is much harder because at this point we have these 500 public data sets, but they are not having necessarily quantified amounts that would allow you to query one study against the other study. And if you do that, of course, you then would like to compare different studies, and you would like to learn about these studies in terms of pathway enrichments and so on, which is yet another animal that we have to treat with. Okay?
Yeah.
So in terms of the Mass Bank of North America, what we really try to do here is in using MS-Dial, which is a freely available tool for PCs, so it's not for Macs, you can upload your spectra. The Mona will then do autocorrelation and scoring, and the scoring is based on the number and quality of metadata. For example, you can tell us a lot more about the chromatography, about the solvents, about the mass spectrometer you've used, the internal standards, and so on. So that would all go into the scoring of the quality of your upload. And then, a user can now do many different types of queries and then download the spectra as well. And here you see just one image of how you can use MS-Dial, which is highlighted export to Mona, and then you can select a specific spectrum, upload it, and you get also an automatic annotation that this has been uploaded, which you can then go for your publications and says, yes, this has been uploaded to a public database. And that's the idea that then later on people can tell that to journals when they report a study in journals. They said, yes, my novel compound has been uploaded and it's now in the public domain. So in terms of downloads, we try to have it very easy. We do allow different types of downloads. Most importantly, the MSP file that can be used in various software, including the NIST MS Search, but also an MZMine or MS-Dial or any other type of software tool. But we also enable MZML data formats that include a lot of metadata in that. So here's just an example for to visualize it, how such downloads can be used. So it is important that also the community now realizes there's more metabolites than just glucose and fructose, and that's where we meet with the natural products community that is represented here by Pieter Dorrestein, and also represented here by a natural product that is asparagine, which we may find even in human samples, because humans eat plants and humans have lots of microbes. So we should learn that the metabolomics chemical space is much larger than just the space that is given us to by the KEGG database.
So for me, one of the key things that we have to contend with is reproducible data analysis, and I think this illustration here highlights it. You know, somebody did a custom analysis, they had an R script for it, and then it doesn't work. And I think we all, many of us, have seen this kind of scenario. Funding bodies are beginning to care about this quite significantly at the moment. Certainly NIH, but also NSF, and even some of the foundations are really beginning to think about it. So when data analysis infrastructure and computational tools are being developed, we're going to have to keep this in mind. In our case, in our GMPS infrastructure, GMPS is basically our tool where you can upload any metabolomics data set, and we have I'll highlight later, a continuous analysis in there. But essentially, every search, any manipulation that you do within our infrastructure is remembered in the way that Google remembers everything any other user does. But by seeing how you do your analysis and remembering your analysis, it also improves reproducibility. And so we have two areas. One is in my user space. Those are the data sets that you subscribe to or data sets you've uploaded. Those are data sets that you're really interested in. And under the jobs tab is any search that you've done, and any parameter that's associated with the search is remembered. Even the current status of the database is remembered at that time. And these can all be shared. And so here's an example of a search that I did. And then you can look at all the library hits that you've gotten. You can create a network. There's some other analysis that is remembered. And then you can hit clone in the upper left-hand corner or the middle left-hand corner. If you want to reproduce and maybe make a slight alteration to the parameters, but with clone, you're just going to reproduce all the settings that you originally had with any search that you did within our infrastructure. And I think this is just the first example of how databases can serve also as a way to promote reproducible science. And I think the metabolomics community at this point would benefit significantly from this, irrespective of the type of analysis you're doing, whether that's heat map generation or any type of other generation, but the memory of how everything and all the data analysis that went into it was done, so that other people can go to your analysis workflow and say, oh, well, I'm not so excited about this component, but this component of your analysis is really good. I should reproduce that in my workflow or not, so to speak. But in the end, you know, we get a table of molecules. If you're lucky, you get a table of annotated molecules, but in general, we get a list of features. If you do an untargeted metabolomics experiment, then that just doesn't make data very informative. And so I always highlight that visualization of data is very important whenever you deal with large volumes of data. And here's a really nice example just to illustrate why data visualization is important. And this really is important if you're going to develop computational methods for metabolomics data analysis. You also need to visualize, because if you don't visualize, you cannot interpret the data, you cannot build a hypothesis, and you cannot really essentially not do anything with it. But let's say that, consider that those numbers that you see in the left-hand corner. This is from a SciPy meeting presentation that I grabbed. But essentially, if you squint, maybe some of you can see what's going on there, but I can't. Even if I give you the heat map, it's still not very informative to you. But now if we translate those numbers and actually replace them with colors and show it as an image, now it becomes interpretable. And this is true with any type of data. That we can look at all the tables that we want, but it's not informative as a way to visualize it. And unfortunately, there's lots of people beginning to think about it. Both myself and Oliver think about visualization of data all the time, and I'll show you a couple of examples. Because I really think that data visualization of chemical data is critical to understanding. And so molecular networking is one of the tools that we have that we've developed for visualization of chemical data. And basically what molecular networking does, it finds relationships between related spectra. So here we have a set of spectra that were based on computational methods deemed to be related. It's called molecular networking, the approach that we're taking. So if you look on the left, you'll see two spectra. They're very similar, but they're 12 Daltons apart. Because we have reference library, the red nodes are part of the reference library. The other ones are in the data, the orange ones are in the data set from a fungal data set that a user had uploaded to GMPS. But you might say, okay, 12 Dalton is carbon. Where does a carbon come from? Well, let's look at the molecule. So here we have this tetraspiralide type molecule, and then the related molecule is this molecule. If you look up in the right-hand corner, you'll see that there's a difference between a methyl group and a cyclopropyl group. That's where the 12 Dalton comes from. And so, but through building these spectral relationships, you very quickly know that these are related. And so if you can annotate one of them, you can often propagate these annotations. Here, for example, you see a 2 Dalton difference. Well, that's if you look at the left-hand side of the molecule, there's a double bond that is missing in those molecules that were present in the molecules on the right, and that's where the 2 Dalton comes from. But in many ways, you can begin to propagate. So here in this particular data set, I didn't highlight all the molecules, but there are 16 potential novel analogs that are structure related to these molecules. And so if you look at the orange node where it says 14 Dalton, we know that's a 14 Dalton difference. And if you very carefully look at the mass spectrometry data, you can probably begin to interpret what region of the molecule has this 14 Dalton difference. And so you can actually begin to propagate some of that known chemical knowledge that we have into the unknown space, even into space that remains completely unelucidated. But there's lots of really great ways to visualize metabolomic data, and people are working really hard on it. So Oliver has this really beautiful GCMS-based fragmentation classification of organic acid, amino acids, etc. And I would really recommend anybody to look at these. I think it's, quite frankly, I think it's one of the major advances that has happened in metabolomics in a long time. Brian Bachman, they have self-organizing maps. These are very nice. Gary Piata and Gary Schutz, they have these bubble plots that highlight, the molecules that are up and down regulated and the significance. Sebastian uses fragmentation trees to begin classifying the kind of molecules they are, whether they're peptide, sugars, etc. And these are all very important ones. Of course, we shouldn't ignore the KEGG pathway maps, but I think Oliver alluded to it. If you do a targeted metabolomics experiment, in my hands, less than 1% of my data really typically falls into these type of pathways, and most of it I cannot yet annotate. And so while useful, and this is the biology that most biologists have been trained in, it also provides a limitation, but on the other hand, very interpretable. The other type of visualization that we have been working on very hard, as well as other people in imaging mass spectrometry, is basically spatial analysis of molecular information, because then you can build relationships of chemistries with other types of information, such as sequencing or activity. So here's an example of our human maps, where we have the idea that if you collect chemical information spatially, you can then map it back onto those locations, and through that you can provide interpretations. And when we published this, Francis Collins highlighted that this is actually a new way to map big data. It actually extends well. These type of tools extend beyond the metabolomics infrastructure, but it was built for metabolomics. But it allows you to compare, let's say, bacterial data with molecular data. Sort of getting closer to the conclusion, and probably the last few minutes before we go to the poll and then the question and answer session. I think both Oliver and I feel very strongly that open source and accessible knowledge is the only way forward. I think the databases that are going to be closed, all that knowledge will be captured very quickly, and they will go extinct because of it. In the same way that Tranche did in proteomics. Tranche in proteomics was the proteomics repository that really didn't see much use, and the data that was deposited there never saw any reuse. But we learned a lot of great lessons from this, but let's not repeat those lessons or the mistakes that Tranche made in metabolomics, and I think we're well on track for a much more positive outcome through integration of all the different resources that a lot of people are beginning to talk about now. One of the challenges that we face is that there's no score of quality of an assigned molecule in metabolomics. So how do we know a match is really a match? And this is true even in targeted analysis. For example, if I say I have a hexenoic acid, a double bond, retention time and the mass are probably going to be the same irrespective of where the double bond is. We may not be able to separate cis or trans out of that, even if the MSMS matches. So there's still no quality score. Ultimately, we need to begin thinking about if data deposition for metabolomics is going to be mandatory. I mean, right now, I saw about 500 to maybe 600 projects that are actually in the public domain, which is, I personally think, not very many. But if we're going to ultimately require people to upload and deposit data, we shouldn't do it at the end, not when we publish, but earlier than that, which means that people should get more benefit out of making depositing data than not. We need to begin thinking about how we can improve the reuse of metabolomics data. I think one of the ways to do reproducible science is that when publishing, we should have a hyperlink with results and parameters of search, or at least some sort of downloadable format that, such as that Mona is providing, that allow you to really know how has the data been handled up until the point that you published. There are guidelines for reporting metabolomics data, but those current reporting system doesn't take in account such as sequence information. The current guidelines are in many ways insufficient for this, but they are a great start and a great dialogue to have in the community. What are the reporting guidelines that need to happen within metabolomics data? Because I've gotten papers to review that tell me that there's cocaine in potatoes, or the crown ethers, which is a molecule that's used in organic synthesis, the crown ether, in microbial physiology because they got a metalin match for it, for example. So these are the guidelines set for reporting metabolomics data may limit those kind of challenges or glaring obvious errors. How can we begin linking the different metabolomics resources? I think GMPS and Mona are beginning to do this, but both of them don't do it adequately enough, and this will require the community, in my opinion, to get together. In the end, what we want to do is, can we pull out all the chemistry that can be learned from a single MSMS vector? So if you have 1 million MSMS vector, for each one of those MSMS vector, we need to pull out all the chemical information that we can get. And it should be much more streamlined than we do this. But one of the key questions is, can we begin to predict spectra? So a lot of molecules will never isolate, or they've been isolated in the past, and it would take such a heroic effort to get an MSMS spectra. So I think it's really important that we begin to accurately predict spectra. And so I think Oliver will take this over from here.
So can we predict spectra from structures? So, oops, that was fast. Okay, so we have done this. We've published last year a paper where we did the prediction of lipid spectra from lipid structures. The lipid structures, we used a template where we said it's a scaffold. We had a glycerol backbone, and we had a head group, and we would have the acyl groups onto it. And we generated this these spectra, not only the ions themselves, but also the intensity of those ions in different instruments for the ion trap as well as for the QTOF. And so we have publicly available over 200,000 MS spectra in positive and negative mode for complex lipids for 29 lipid classes. So the rules were done from authentic spectra, and then we took about 3 years to fully validate those predictions. That is Tobias Kind's work. And you know, from other types of reports, from journal reports, because there was no database where we could basically download it, and it showed that it can be done. So there are many more prediction software programs out there. CFMID from the Wishart Group, Magma from Netherlands, Midas, Medfrac from Stefan Neumann, Mars Frontier, which is a commercial software, MS Finder that we will publish with our collaborators in Japan, and FingerID. And we have started asking the question, you know, how valid are these predictions? You know, how can these tools be matched? So we used 1620 spectra from Mass Bank, and this is work by Tobias Wermuth, who's an intern in our group. These are all accurate mass spectra acquired on an LTQ Orbitrap with, of course, annotated structures. So we took the structures, we predicted the spectra from those structures, or we tried to match the experimental spectra to those, and experimental structure to the predicted spectra. And when we ask how often, on average, would each software annotate the actual ions that were found. And you see this on the slide on the right-hand side. You see for about half of the ions that were found, CFMID would correctly predict those. And you see for other software, it would be lower. And then we said, okay, so that's a start. And we asked, what is the overlap? You know, would these different software tools predict the same type of ions or different ions? And if you look on the bottom, if we use CFMID from the Wishart Group on these 1600 compounds, of course it has, overlap 100% to itself, but it's a very, very small overlap to the other software. So Mars Frontier, for example, would have only 3% of those ions that are predicted from CFMID would also predict those, whereas Mars Frontier would have a larger overlap with Magma, Midas, or Medfrac. If you look at Magma, Midas, and Medfrac, among themselves they use similar algorithms, so they think at least they have a similar overlap, 50 to 60% overlap between those tools because they have similar algorithms behind it. What we learned from this exercise is that over 40% of all the ions from these known compounds were not annotated by any of the software that we used here. So we are not there yet for accurately predicting it and using those on a broad scale, but we have done a start as a community using these different types of tools, and we also see that these different types of tools come up with different types of ions and different type of predictions. There is a lot more validation work that is needed there to see where the rules fail, where the software fail, which algorithms would be good for which types of compounds. And I think this is a massive undertaking that we have to do in the next 10 years. But I think also in terms of the universe and the complexity of 100 million compounds, this is the only way forward if we really want to conquer the metabolome together, apart from, of course, getting more experimental spectra and the molecular networks that Pieter Dorrestein told us about. Okay.
Yeah, so that gets us to the last question is how do we share metabolomics knowledge? And I think we've seen certainly reference libraries. But what about if you come in with your data set and you want to be able to learn about your type of information? And I think we have some opportunities in the metabolomics community to do this much more efficiently, and I'll just show you one concept of how knowledge can be shared among projects, and that is something that we've introduced within our global natural product social molecular networking, and that is if you have a data set, the knowledge that is accumulated in the metabolomics community should change constantly, and it should continue to grow constantly, and can we capture this knowledge with your own data? And so what we have done is essentially what you can do is you can upload your entire project. So we had, for example, somebody in Hawaii uploading 16,000 LCMS runs, but they were annotating their information, and so meaning they were annotating their MSMS libraries. And at the same time, we continuously search against NIST 2014, MassBank, Respect, HMBD, GMPS, all the other ones where we have MSMS libraries accessible. We will search against, and in the future, just the same way that Oliver is doing, we're going to probably incorporate in silico generated libraries from all these different sources as well, including their lipid blasts, for example, so that we can update the community what knowledge exists within your particular data. But let's say you work on a project, and let's say you annotate an MSMS spectra. We had somebody who was working on an algae invasion in coral. They were annotating data. And then we got an email saying, hey, you have new information, and this matched up to a data set that we had created during an exacerbation of a cystic fibrosis patient, meaning something in the corals during algae invasion was similar to what happened in a patient, in a person. It turns out that corals have an ancient immune system, one of the oldest innate immune systems, and these were inflammatory lipids that were identical during these kind of scenarios. And so that's how you can begin to share knowledge. So if any new information that you annotate, if that matches to anybody else's data, they get an email saying, hey, there's an update to your particular data set, because behind the scenes we cross-correlate every MSMS spectra to every other MSMS spectra that is uploaded to the infrastructure. And so you really get this community-wide learning, and this is just one way that I can see how future knowledge about metabolomic data is really shared among the community. I think we can have Amazon-type prediction of what data sets you should be looking at. I mean, there's lots of different ways, creative ways, that the community can really benefit from having all this information readily accessible. The other thing, within GMPS, you can subscribe to any data set. So if you are interested in plants, you can subscribe to all the plant data sets. If you're interested in actinomycetes, if you're interested in mice, if you're interested in human cell lines, cancer, whatever, you can subscribe to those different data sets. And if new information becomes available, you'll get that information relayed to you about those particular data sets as well. So this is one way that you can come together as small communities to annotate your own infrastructure, own data sets, or share with the worldwide community. In this case, there are already well over 6,000 users within our infrastructure that are beginning to take advantage of the concept of living data. But if all metabolomics repositories, infrastructure, databases were connected in a way that facilitates this type of sharing, and I think it's emerging, at least for several of the databases, this is going to emerge. It will be easier for anybody who does metabolomics, untargeted metabolomics, to capture the knowledge that's out in the community, rather than having to go and do Google searches if you have some sort of M over Z feature that you're interested in. In our case, you can see the growth. It's been out for a year, but we have a 22-fold increase in spectral ID. But of course, you need to ask, well, what about quality? Of course, unfortunately, there's no scoring function that tells you whether a match is really correct or not in terms of probability of being correct. If I say it's glycine or azithromycin, is it really azithromycin or glycine? So one way we've dealt with the quality is actually going back to the community itself and provide quality rating from the user community, where 1 star is an incorrect identification, 2 stars is not enough information to tell, 3 stars is an isomer, possibly correct, and then 4 stars would be correct. But this is just a way that knowledge can be captured, and so if you have a lot of if you have information where a lot of people say, well, that identification I don't really trust, you know, that knowledge is now accessible to the user as well. And so this is just one way to deal with databases and computational infrastructure, and it's sort of a forward-looking vision of what metabolomics is going to look like, I think, over the next 10 years. And then finally, thanks to the different people that are on this slide, and I think we will go to the polling questions now, and then question and answer session.
Yeah, thanks to Gerd Wohlgemuth, Sajan Mehta, who made the Mona possible, and Hiroshi Sugawa for the MS Dial, Masanori Arita for the original mass bank.
And thank you guys for the presentations. Dan, can we go ahead and move directly into the poll questions now?
Sure, we'll do that. So,
do I need to make you a presenter, or will you just, it just pops up automatically?
We'll see. I think it's fine right now. So,
yeah, there we go.
All right. So, first question is, what is the most pressing issue in metabolomics databases? So here you get one choice. A is at the number of authentic compounds in mass spec spectral libraries. B, the number of authentic compounds in mass spectral libraries. The structure of diversity of compounds in current mass spectral libraries. D is the annotation of positively identified compound and species, so species metabolite databases. And E is the association of compounds with biological function in different species or diseases.
Yeah, I think there was something wrong, but...
So if you would...
Okay.
The first two look to be repetitive.
Yeah, the first two look redundant, so we should pull them together. I think that I wanted to ask some other question.
Okay. Well, when we get the stats, we'll be able to tell.
Yeah. Okay. About 60% have voted, and we have nearly 70 attendees, so you could cast your vote. Come on, we need to get up to 80%.
So this information is very important to us. It provides quantitative feedback that we can use to better assess the consensus of the community.
All right. All right. Well, that's 77% of the community that's voted, so I'm going to close that poll.
Great.
The next question is, which mass spectral repositories have you used? And here you can select more than one response. Okay, we're at about 75% response rate. Oh, we're going to hit 80, I bet. There we go. 80% have voted. All right, I'm going to close this poll in 3, 2, 1, close. And then the third and final polling question is, which mass spectral prediction or matching tools have you used? And you can select as many of these as you've used in the past.
Oh, what is missing is none.
Right.
So it seems to be a split across several different ones of those, the percentages. So 30 to 40% are using NIST, MetFrag, and others. At the end of the presentation, we'll provide...
No, it's not limited. It should be one or more. And so 35% have used NIST MS.
That is correct. So it's and 35% of the pollees are using MetFrag, and 40% are using others.
I don't see that answer.
It's under organizers. You should be able to see a polls tab, and it shows this in real time. Yeah, we'll come back to it and give you provide the feedback.
That's great.
Okay, we're going to close this question in 3, 2, 1, close. All right. All right, so let's look at the, I'm going to share the results here so everybody should be able to see this. What is the most pressing issue in metabolomics databases? Okay, so the first two, I guess, were ambiguous, so that's 43%. Say the number of compounds. The next largest one is annotation of positively identified compound in species or species metabolite databases seems to be the next biggest response. So, all right, any discussion by the panelists on that poll?
I am surprised about this 33% species to metabolite annotation. I did not know that this was such a high ranking.
Yeah.
Yeah, it's a question I often get, actually. So I think that maybe we should begin thinking about capturing that knowledge.
No, absolutely. I think it's interesting.
Yes.
All right, let's go to the next poll question. Which mass spectral repositories have you used?
And there's a note from one of our members that we didn't include MetFusion.
MetFusion and MetFrag are almost the same.
Yeah. That is better. Well, her comment was that it's better than, or the person's comment was it's better than, so MetFrag.
So the mass spectral repositories for GMPS, since it's not published yet, it makes sense that only 14% have used it. But on the other hand, 14% is actually a large number if you consider it's not published.
Well, you can go to GNPS and download and look and so on.
Mm-hmm.
But in a way,
it's not in Google yet.
Yeah.
It's not in Google, that's right. You have to know it and then you can find it.
Yep. So for the people
let's
who don't know, it's gmps.ucsd.edu.
Okay.
All right, let's look at the last poll question. Which mass spectral prediction or matching tools have you used? And a lot of people use other. I don't know what that means.
Mm-hmm.
Yeah, I think it might mean none of them, right? Because if you want to poll, if you want to participate, but there's magma and whatnot, but I didn't want to put all of them there because many of them are not very well known.
Yeah.
Actually, for me, this is actually nice to see that people are beginning to use it. I've seen limited publications that have actually really used them to identify specific molecules. But I'm excited that these tools are being developed because I feel that this is the frontier of, one of the frontiers of metabolomics at the moment is how do we in silico begin to predict the molecules. So this is pretty exciting.
All right. Okay, so we'll hide all the poll results. And now we're looking at Pieter's desktop.
Okay.
Again.
Yeah. You might want to pull up your slides again, Pieter, and as we begin to go through the questions, you may desire to flip through those.
Yep.
Okay, so
Dan, while you're pulling up the questions, I would like to start with some of the questions. And I've got a series here, but I'll just ask one until you can get those questions ready for our presenters. So the first question I have is you talked, both you talked about the importance of databases and communicating between databases. But to do that successfully, there has to be some level of standardization of the data and the data format. Do you think that we are where we need to be there, or what do we need to do to get that data standardization compatible across databases?
I have strong opinions on this. I'll be interested to see what Oliver thinks. There is some needs for compatibility. I mean, certainly within our Chapter, we take MCML, MCXML, .mgf, as well as most of the vendor formats, but it's actually really, if you look at the conversion tools that are out there to a common format, it virtually, many of them make mistakes. Let's put it that way, very gently. And so from a data standpoint and data format standpoint, I think it would be very useful if we indeed have conversion tools. However, everybody has their own preferences, which means one of the things that would be beneficial if we have the vendors tell them at least, we need to have good conversion tools that allow you to get into these kind of resources, and they're willing to often work with people, but they don't realize how important it is.
Okay, so talking to that, I would like to say that some original data, some vendors are a little better in terms of conversions than others, in allowing that already from the vendor software. Yep. For those people who don't know, there are also tools available from the other community. For example, for data conversions and mass spec conversions, ProteoWizard is a real nice tool that's coming obviously from the proteomics community. But since it's just mass spectra, it can be easily converted and easily be used, and people should know about it. There's DataBridge from others. That's one thing. In terms of the standardization of structures, just the fact that we are able to link into these different databases and use them, HNBV, GNPS, NIST 14, that already means that at least from the database community, we have already standardized in terms of spectra, in terms of compound structures. That has been achieved. Now, what has not been achieved is the metadata standardization, right? So the how do we talk the same language when we say accurate mass? How do we make sure it's an accurate mass? What do we mean by an accurate mass? How accurate is it actually? And how do we get ontologies on the different types of instrumentations so that we can tell, like, okay, I want to have all types of traps. Give me all spectra that have been generated by a trap, or give me all data that have been generated by some type of QTOF, or whatever name a triple TOF is from another vendor, right? So that is something we still would need to work on, and I think that is possible because there's only a limited number of those types of metadata. That's what we are working on right now.
Yeah, so that's the metadata associated with these with the actual spectra. One other way is the quality of these annotations. All libraries have pretty much mistakes in them, and so one way to change this is to have dynamic updates where the community can indeed provide community annotations. We've built something like this in GMPS, but I think other Metabolomics will begin to do similar type of things. So there's three components to that question. Really, the data conversion, so data compatibility of the data that gets collected on the instrument. There are reference libraries, but then also when you get into data analysis. In the end, what we often get out is a list of features, MOZ, retention time, or MS2s with retention time, and so on. every analysis tool or generates its own table format. And I personally think that should not be standardized. Partly is because it's easier to develop a script that allow you to convert one table into another to be compatible with other analysis infrastructure. And I think there are so many people that are going to develop these type of analysis infrastructure and table generators, so to speak, to allow pathway mapping, to allow molecular networking, to allow spatial mapping, that it's going to be really hard to even get the community on board for one format. And so I think developing scripts between the tools, much the way Oliver described in the beginning, that you have, it's okay that you have different databases and different resources, but that you can somehow create a link between them. And for me, if you develop scripts for conversions, that's the way to begin linking them. And that also allows you to link to outside tools, let's say the microbiome field, other statistical tools. You can go to architecture tools that are not designed for metabolomics, begin using them because you have different table formats that can be converted into the right format, so to speak.
All right, thank you, gentlemen. Dan, can you go to the audience questions now?
Sure, I'd be glad to. If after I make a point or a comment, if you would like to speak, please use the raise your hand button, and that way we will be able to unmute you and you'll be able to interact. So Tagreed Al-Shufani made a comment. Could you please repeat the software tools which were mentioned in this talk and which ones are freely available? Maybe the question could be, or it could be rephrased, are there good places to look for what's currently available, like comprehensive lists of software and so forth, and databases?
Let me see. There's a recent paper that came out. It's the closest one. So this is by Bernd Lang. It's open access metabolomic databases for natural product research. That's one place that you can go to for providing a list of these things. Oliver's website has a lot of them, of the tools that he has created. So there's no, as far as I know, not a centralized list. This is the database that I have. You can take a screenshot of it if you want. But for uploading data and projects, you have Metabolites, and as far as I know, Metabolomic Workbench and GMPS. Maybe there are other good ones that the rest of the community knows. Oliver, maybe you have some. And then there's reference spectral databases on the left.
Yeah, and what we have not talked about is, of course, chemical databases like ChemSpider, PubChem, ApeGMDB, whatnot, right?
This is just raw metabolomics data.
Yeah. So for raw metabolomics data, and for the projects, I think Metabolites and Metabolomics Workbench is a good way to start. And the NIH really wants also people to use those and to give feedback. So what the NIH asked us to do is to disseminate the idea that people should go to these repositories, download studies, and report back what they liked or what not liked, so that we can ask those contributors, which are actually in San Diego, to improve how data are presented. Mm-hmm. And the same thing is, of course, then true for Mass Bank of North America or other types of repositories where we are always looking for feedback of what people like. And I don't think there's one unique repository for the question of matching spectra. And we have written a recent review this year in Trends of Analytical Chemistry, I think it was. So that's my name and Trends of Analytical Chemistry, where we put in a review on that question.
So maybe the question brings up the idea that there would be value to putting together somewhere a listing of these different freeware or software and databases that could be made publicly available.
Reza Salik had a hand raised, so let's see what he wanted to chime in on. Reza?
Hi, hello. Can you hear me?
Yes.
Yes.
Oh, it was a wonderful talk. Thanks, Dan and Lloyd's, Oliver and Peter. It was wonderful listening. I was very interested. Many notes and many questions. We had similar discussions and ideas of federative distributed databases. So instead of everyone reinventing or partially reinventing the wheel, will be to use advantage of what in a database or a resource has been done quite nicely and avoid others repeating. Other omics or sciences, not necessarily metabolomics, to an extent are working, excuse me, and have done it. But it has certain requirements. It's in terms of being a nice way to be able to incorporate resources to one another, basically being able to communicate via certain APIs and tools with one another, computational requirements of it, and it's all a work in progress. So I wonder, what is your opinion on these things? And secondly, was also, so the total number, the 600 number, partially 100 being the within EBI resources, that sort of I'm from EBI, it's very, very poor of data sharing. I think we started from 2012, all these resources to capture metabolomics information. And if you consider the past 3 years, we mainly represent 1 or 2% of, I'm very optimistic on all the entire data being produced, or maybe 5%. Compared to other omics, this is very low. We try to encourage via journals, via resources, to increase that, but partly the burden is slow and partly it's manual process of submission. It requires ways to automate it. So I wondered, what is your opinion on that as well?
So I have an opinion for data uploads, maybe why it's not being done so effectively. One, unlike sequencing, where it's mandatory to have an accession number, there is no such thing for metabolomics, and I would be shocked if we are going to get the community and the worldwide community up to that level. Sequencing started at a very different time. And so we need to come up with a more creative way of people making data accessible and actually seeing the value of it in it. So what this shows to me, in reality, it shows to me that the community in general doesn't think it's valuable to have their data accessible to the worldwide community. And so how do we change this? I think as metabolomics community starts to think about it, we need to make data upload part of the workflow so that when a person uploads data and does the analysis, they get maximum amount of information back. And then in terms of cross-sharing, yeah, ideally if the major tool developers for metabolomics, where Medlin, HMBD, MassBank, if they develop sort of API interfaces that allow you to communicate from one tool to another, I think this would be really wonderful. And the reality of it is some people will not play that way. Other people are going to develop tools that are going to share. It's the same way that some people like to publish in open access journals, some people like to publish close, and that's fine. We should have room for both, in my opinion. But I think there's a larger and larger community that is excited about developing tools that can be shared through API, at least that potential. There's a workshop that's going to be organized specifically on this topic in a few months, where there will be 30 to 40 people that are going to get together and discuss part of this issue, not an issue, but part of this exciting development in metabolomics.
Where is this workshop?
It's going to be in Germany, but it's a fairly close workshop initially, and later on we're going to plan on opening it up in a larger scale.
The Dark Stool seminar?
Yeah.
Oh, okay.
Okay, let's go to another comment point. Robert Mistrick made the point that MZ Cloud, which is the third largest library, is missing. So, Robert, did you have a question or a comment?
Yeah, I have a reference spectra. It's on the bottom here on the slide that's up.
First, I would like to go to these resources. On the Metabolomics Society website, there is a page which lists all databases and software, so it's very comprehensive. And second, what I meant was this pool, because the MZ Cloud is now based on the Spectra, I think the largest reference database.
Yeah, but you can't download it. It's not accessible to the community, so you cannot develop computational approaches.
Well, yeah, it's true.
So and that is, of course, a problem a little bit. Who pays for these repositories and for the development of these repositories? Because that's the reason why some of them, like NIST 14 or Metlin, are just not freely available, because they are either paid by government and then they're distributed by license fees, or they're paid by sponsors like Agilent or so. And again, they would then require that these are specifically not like freely to download. And you know, on the other hand, the agencies would not pay for spectral repositories by themselves, and they also would only have a limited funding available for making repositories like the Metabolomics Workbench, where they hope that eventually this would be not requiring a lot of curation and no real data money input anyway. And this is always a problem in many of those repositories. How can they be maintained, curated, and over a long period?
All right. So we had one more comment maybe we can address, and I think Laura, it's from Laura Reed, and I believe she's disconnected, so I don't know that she can. If you're there, Laura, please raise your hand. But she made the comment or question: do species-specific metabolomes really matter? If the chemical IDs are good, isn't that enough independent of species? So maybe some comments from the panelists on that.
Well, as Pieter said before, we don't have a really good assessment of the quality of matches. And when we do, we use, and we have discussed this before, you know, about that's this paper by Aaron Krieg and others in Metabolomics Journal, where it says, how confident are we when we say a certain spectrum matches another spectrum? And Pieter said, you know, you would have cocaine in potato. That doesn't sound very likely. On the other hand, who knows if, you know, if there's not some weird enzyme that would make cocaine? I mean, I don't think so, but, right?
We would be very rich.
Very, very weird. So there is this, you know, the species and the authentic knowledge about that this species really makes this compound would then enable, in fact, making pathways and predicting gene functions. And Ji Lu in China has given a very nice talk at the Metabolomics conferences here, where he you know, he did it for flavonoids and other natural products in plants, and then said, well, once we have found those and we really know that these compounds exist in my species, in my germplasm, in my GWAS population, now I can look into the genes and associate the functions of those genes, and then can validate those functions. And he had nice papers in Nature Communication and PNAS and other journals where he showed that principle. And I think, therefore, it does make a difference to know that a certain species or in a certain organ a compound is made or not made, even for the gene function annotations.
Yes, I totally agree. I think there's very There's a lot of possibilities for gene discovery and better understanding of chemical diversity and genetic diversity as we begin to look at the metabolomes and genomes of different species.
Yeah, I think this is long term. I think this is really valuable because so in GMPS we can now quantify the diversity of MSMS spectra of all the community data sets, and we have nearly 1 billion spectra in there. 200 million or so are unique. So when you narrow your search space down, because you're searching against a specific species, you're less likely to get false positives or false negatives, or, well, really false positives. And so this is where species identification becomes very important, because if I get a surfactant and I'm studying surfactant, which is a Bacillus solus molecule, and I'm studying urine from a mouse, you know, that doesn't make sense. But if I get surfactant and I get a Bacillus species, and I'm studying a Bacillus species or some random microbe, you know, then the potential is much higher that that's correct. You know, how to quantify that, I don't know, but we won't be able to develop quantitative or probability statistics unless we have that species information. So I think there's value in it. I don't know truly how to capture that all the time. Maybe we can mine PubChem, ChemSpider for some of this information. Maybe not. Maybe we can have the community tell us, okay, what species have you found this in? There's ways to capture that knowledge and retain it.
Okay. Let's see, we have one more question. Are there any best practices for creating and documenting metadata workflows that could help lead to standardization? That's from Carl Leonard, so I'll unmute you, Carl. Okay, you should be unmuted.
I don't think so. I don't think there's really good workflows for metadata. Anyone?
Nope. I mean, we're trying to create some within my own laboratory even, and that's already pretty challenging. We can look at outsized resources, so I think Chime, Cheetah, which are the metabolomics, microbiome workflows, they have somewhat of a standardization method for capturing metadata, but they do it even before data is acquired, capturing some of the metadata. That's a part of their input of analysis workflow that is needed, otherwise you can't analyze your data.
In some software for the mass spectrometers themselves, they would at least acquire and disseminate the metadata on the electrospray voltage and the fragmentation voltage. That type of metadata is done by the instrument vendors usually, and therefore it can be captured. But of course, anything else like, you know, how you treated and harvested your sample and so on, that is usually covered in LIMS systems. And again, these LIMS systems are not standardized at all. They are laboratory specific. That's why they're called laboratory information management systems. And I don't see, we also looked at different open access LIMS systems and see if there's anything useful that we can just download and use. And those usually didn't work. So that was a little bit of a pity.
So maybe we should begin to look at some of the other areas. So the microbiome community has somewhat come together and said, okay, this is a data form or a format that captured metadata that many of them use, but not everybody. You're always going to have people that are outside of these kind of things, and I think that's fine. Yeah.
Okay. All right, Lloyd, I think that's, I don't see any hands raised or any further question.
Well, I have several still, so if we have about 11 minutes left, so if I can indulge. The point was made that visualization of the data is very important, and the graphic you presented, Peter, was very impressive, and it really did press the point across. So I stop and I ask the question, is this another grand challenge that we face in metabolomics? And if so, maybe we should put this into our series as well.
Oh yeah, I mean, I think so, because even in plants, of course, it's cellular organization of metabolism is critical in plants.
Humans everywhere, it's important, I think. I think it would be really good to have a grand challenge on this.
Okay, I think so too, and I think that we'll look for volunteers of working that in too. Pieter, you also made a comment that the reporting guidelines for metabolomics are insufficient, and you suggested we look to sequencing for greater guidance. Could you expand on that?
Not necessarily sequencing for greater guidance. The key part was that, for example, doesn't take sequencing in account. Sequencing can actually provide you pathway information which can be organism specific. It can, if you look at Michael Fischbach's cell papers, who defined all the gene clusters that you find that make specialized metabolites in microbes, there's so many of these are unknown. I would say less than 1% of these are known structurally. But on the other hand, you can make some reasonable prediction about structural class and that type of information. And that type of information can give you higher confidence about the assignment of the chemical information that you can collect by mass spec. And so that's what we, but that type of option is not yet incorporated in the guidelines of how we should report metabolomic information, if that makes sense.
Yeah, I'm still trying to figure out the whole story there. So what I'm hearing you say is through GWAS type of associations, we can better predict metabolite structures. But that's dependent if we have sequence information that's co-acquired with the metabolite data. That's not always the case, and actually at many points it's not available.
No, exactly. So, but on the other hand, metabolomics is so diverse. But on the other hand, any chemical information that we can get, right? So GWAS, you can do co-association, which is great. Microbiome sequencing, if you do metagenomics, you'll get pathway information that are responsible for making specific molecules, isopreneurs, polyketide, etc. That type of information you can sometimes use to assign, what is the dominant structural feature of a molecule. Mm-hmm. In some cases, you can actually use it to really predict the structure and then match it up. So those are the kind of pieces of information.
Sure. So I understand that we can gain information that way, but thinking back to the statement about reporting guidelines, how do we? What do we need to do to the reporting guidelines to account for this type of information that you feel is missing?
So what are the key pieces of information to get to the structure of a molecule? So if you have a spectrum and you say it is this likely this molecule, you have different levels. You have unambiguously identified, which I'm still wondering what that sometimes is. Because even if you do retention time alignment with an authentic standard, and the example that I mentioned was a double bond on a lipid, they may have nearly identical MSMS spectra, they may have MS1, and the retention times are the same. Is that unambiguously or not?
There are papers. There are reports where people won a Nobel Prize and later on was discovered that the structure they assigned was not correct.
Yeah, but I'd like to start. So what the guidelines don't have the ability yet is to take into account some of that additional information beyond what's collected with mass spec, and sequencing is the example. But what was captured with NMR? Sometimes you get partial knowledge by NMR if you have sufficient quantities. If you don't, maybe there's a UV trace that is associated with it. But the one that is really hard and is very useful at times, at least in my hands, is the chemical structural prediction that exists within genomic information.
Mm-hmm. Okay, I think I understand now. There have been several recent papers that are suggesting adding quantitative or alphanumeric type of confidence factors to metabolite identifications, and I think many of those do take into account other types of data. They don't take into the sequence information, but maybe that could be incorporated as well.
Mm-hmm.
All right. And then, if I can, maybe one last question, and I think this is more directed towards Oliver, who was talking about lipid blast and using predicted mass spectra in the identification process. And I think this is really a very potentially valuable tool that can really accelerate us to much higher throughput type of identifications. But the problem and limits with that is to build these predicted libraries, you generally have to have good empirical data sets for that class of compound. If you're going to look at flavonoids, you need some flavonoids. If you're going to look at monoterpenoids, you need some spectra of authentic monoterpenoids. So with that in mind, I mean, how rate limiting do you think acquisition of the learning empirical data will be to predicting these, libraries in a higher throughput?
Well, first of all, as you say, it's a great way to go forward. Secondly, the prediction tools are all developed on, or most, at least those that you see there, are all developed on authentic mass spectra that are public. So mostly mass bank spectra. Mm-hmm. So the larger our known chemical space is, the better can we develop prediction tools based on the ever-increasing number of experimental spectra and structures that they cover. It's of course clear if you look outside of those nomenclatures, the prediction gets much worse. So once you go outside of your comfort zone, so to say. And for me, it was very interesting to see that CFMID and Mass Frontier had not much overlap, because Mass Frontier is a commercial tool that uses known fragmentation rules from chemistry or from published chemistry and applies those to those substructures it would find in the structure, whereas CFMID was looking at bonds and learning from the existing mass spectra from mass bank. So it's quite different in terms of approach, and therefore also quite different in results. And if we could somehow make more research into that, I think we merge these ideas, right? In the future, I think that would be very promising. Also to try to get better predictions. What we for compounds that are outside of the chemical space we are right now looking at, or where we have authentic spectra. Now, one thing else is missing here, and this is the quantum chemistry. So in principle, you could say, let's use quantum chemistry because it's mass spectrometry is physics. It's just putting energy into compounds, and that can be in principle predicted. It's just the calculation efforts for these are still very large. But it has been done, and it can be done at least for electron ionization spectra. There is software available to do that by Grimme, the group of Grimme in Germany, who developed that. And this is important to know that this is another way to think about it, rather than using heuristic rules, learning from existing spectra. They're just saying, okay, let's use physics instead. And this has not been not much research yet on this topic.
Yeah, I think one of the things that MC Cloud has now a lot of the predicted spectra, and I think they were done with semi-empirical calculations such as this, or some of these gas phase calculations. I'm not 100% sure. If Robert is still on, maybe he can comment on that. I truly think this is a frontier, and one of the challenges, the reason why these tools or many more of these type of tools haven't been developed is because good libraries to validate the results have not been accessible to the community. I think they're now beginning to become accessible to the community. There will soon be another PNAS paper with a new tool that comes out for doing these kind of predicted values and predicted matches based on the structures that are in PubChem, for example. So people are beginning to think about it, but really from a computer, well, the algorithm developers, they need good libraries to have access to validate their results.
Absolutely. So, Robert's raised his hand. Robert, did you want to make a quick response?
Yeah. So regarding the prediction of fragments between different programs, well, the question is which program actually predicts the correct fragments. And to assess the correctness of the fragments, we started to calculate those fragments in a quantum chemical manner. And now in MZ Cloud, we try to annotate peaks with fragments that were confirmed with the quantum chemistry. At least we calculate the ranking, so which fragment is most likely than other fragments. So because even for mass frontier, it's not a problem to explain 100% of the peak. So, well, the question is how good you explain the peaks. So the percentage doesn't say anything. So the really important question is how well you predict the fragments. And I would like to also agree with Oliver about this conform zone of the chemical space. Sure, for compounds that they have a simple uniform fragmentation pattern for linear molecules, so you can do a pretty good job with a heuristic method like these lipid maps. But once you go to the more complex molecules with the rings and so on, well, I am afraid the only way is quantum chemistry. That's my opinion.
Mm-hmm.
Yeah, I agree. So Robert, will you guys ever be able to make those libraries available, even the quantum mechanical libraries, or is this always going to be behind closed doors?
Well, the quantum chemical libraries, you can preview the library, and regarding the structure, so, well, I was never asked this question. I never expected that somebody would actually like to use these quantum chemically predicted fragments. So, well, as you know, we are associated with a different company, so it's not fully in my power to answer these questions. So therefore, I'm not the boss to decide whether to make it public or not.
Yeah, that's what I said. You know, the money has to come from somewhere, and if you you know, we tried that with the NSF, that we put effort proposals, and then you get rejected, right? Mm-hmm. So if the taxpayer doesn't pay for these efforts, then maybe private companies, and then of course you can complain that things are private.
Mm-hmm. But the question is, can we come up with a creative business model that allows you to still make up? I mean, there's a lot of open access models that are out there, business models that rely on open access that don't limit. So maybe collectively at some point we can begin to think about how to indeed sustain these kind of things with an open access model that supports it.
Well, I think also we shouldn't underestimate the cost. So MZ Cloud is developed by 13 people, including curators, software developers, and so on. So for many years, and you know, the money needs to come from somewhere.
No, I agree. But I'm trying to think: are there different business models that you can that can be developed that still allow the knowledge that you guys have developed to be accessible?
You can buy a subscription, can't you? Is that not a? I mean.
Well, but then you can't. You still, even if you buy a subscription, you can't still do anything with the libraries, as far as I understand.
Mm-hmm. Yeah. So that I think that the ability to reuse the data and is probably the key issue there. So,
yeah.
Okay, I've got 3:33 or an hour and a half into the presentation now, so I think we need to try to wrap things up. I would like to give our speakers one last opportunity to provide some closing comments, and then we'll close.
Thank you.
Okay, thank you. Is that Pieter? Yeah. Anything else?
I think we addressed all these things, and I think we have a very interesting future for over the next 2 to 5 years in metabolomics. There's major changes that are going to implement how people share knowledge and how they communicate with one another about this knowledge that can be gleaned from metabolomics data. So to me, that's pretty exciting.
Yes. Well, thank you again, gentlemen, for your presentation, for the stimulation of discussion, and I too agree that there are lots of opportunities in metabolomics and still some major grand challenges to face. So with that, I'll bring things to a close and wish you the best, and look forward to seeing you all back next month for our next webinar. Take care now. Bye.
Bye.
Muted.